Aloha. My presentation today will be focused on glycogen metabolism, with which I will conclude chapter 15. Now, before I start with glycogen degradation and glycogen synthesis, it is appropriate for us to talk about what is indeed glycogen structurally and where is it located. Now, we know that glycogen is a homopolysaccharide, so it's made out of glucose, but structurally its simplest elementary particle or structural unit are actually beta particles. They're called beta particles, and each beta particle is made of approximately 55,000 glucose monomers or residues with approximately 2,000 branches. It's important to note that each one of those branches is a non-reducing end. This will be important point of discussion later when we start talking about synthesis and degradation. Alright, so 20 to 40 of these beta particles united together form something called alpha rosettes, which are kind of a superstructure of beta particles. Now, where is glycogen located? It is in the muscles and in the liver. About 10% of the liver mass is glycogen, and about 1 to 2% of the muscle mass is glycogen. The glycogen in the muscles, when needed, would last approximately 1 hour, maybe a little bit more, maybe a little bit less, depending on the amount of muscle, depending on the amount of storage, and depending on the muscle, and depending on the need of glucose. In the liver, that storage stored in the liver, or that glycogen that is stored in the liver, can last for approximately 12 to 24 hours. So what are the advantages of having glucose stored as glycogen? When you think about it, why? I mean, it's about the same mass. If you have 55,000 glucose monomers, they will weigh pretty much as one beta particle that's made out of 55,000 glucose residues. So what is really the advantage? I don't know if you remember when we talked about osmosis and osmolarity in chapter 2, I actually gave you that example where we talked about what is the advantage, basically the same question, and why the cell prefer to spend energy to polymerize glucose into glycogen instead of just storing glucose. Well, the reason for that is the osmotic pressure. If remember, the osmotic pressure depends on number of particles, not their chemical properties, not their size, and not their structure, simply number of particles. Well, you can imagine the osmotic pressure the cell will experience if it has 55,000 particles versus 1 particle. The osmotic pressure on the cell will be tremendous if we just have glucose and not glycogen. Another thing is, like I said, the cell will invest or spend ATP to polymerize glucose into glycogen. When does that happen? Glycogen is synthesized as a deposit or a storage molecule when we have plenty of ATP. That happens during a time of plenty. When do we use glycogen? During a time of need. And if you remember, when we talked about glycogen phosphorylase, there is the first step in the reaction after the removal of the end glucose molecules from the glycogen molecule. We're saving 1 molecule of ATP for each glucose 1-phosphate that is removed from a glycogen molecule. So there is also an energy advantage. And lastly, but not the least, it's convenience. I mean, there is a reason why we store glycogen in the muscle. Muscles use glycogen. They use sugar, especially when we have to provide fast activity. And for that reason, that muscles store some glycogen to provide that information, energy when it's needed. So again, osmotic pressure, saving ATP in the degradation of the resulting glucose from endogenous glycogen, and for convenience. So, glycogen breakdown. How does it happen? Why does it happen? Well, you already know the first reaction, which is glycogen. From each non-reducing end, the last glucose molecule is removed in the form of glucose 1-phosphate. What was the name of that reaction? Glycogen phosphorylase. So what's next? We have glucose 1-phosphate. It needs to be converted to glucose 6-phosphate with the help of phosphoglucomutase, because this is an isomerization reaction. And then what's going to happen then? Now we have glucose 6-phosphate. Where can we use glucose 6-phosphate? Glycolysis, pentose phosphate pathway. For energy, what else can we use glucose 6-phosphate for? Well, we can use it to supply or to replenish blood glucose levels so that there will be sufficient amount to provide energy for the brain. Alright, but what needs to happen to glucose 6-phosphate before it enters the blood? I want to remind you again of the glucose transporters that are on the cell membrane. They're passive transporters, and they only allow glucose to pass. Remember, one of the reasons why glucose is phosphorylated immediately after it enters the cell is so that basically to prevent it from coming out through that passive transporter if the concentration falls on the other side. Phosphorylation of glucose to glucose 6-phosphate is one of the purposes, obviously, is to activate the molecule, but the other purpose is to keep the glucose inside the cell, because it cannot pass through that transporter. But now we're generating glucose 6-phosphate in an attempt to transport glucose through the cell into the blood. But these transporters cannot transport phosphorylated glucose. So in order for that glucose to exit, the phosphate needs to be removed. What enzymes would remove phosphates? Phosphatases. This is a glucose 6-phosphatase. Interesting thing about the glucose 6-phosphatase is that it is located in the endoplasmic reticulum, which is basically a space that is carved inside the cytosol, but it's independent. There is transport through the ER membrane, but this area is basically separate from the cytosol. So we need to have glucose 6-phosphate enter the endoplasmic reticulum, then the phosphorus to be removed from glucose, and then glucose and the inorganic phosphate to be independently transported into the cytosol, and then from there to the GLUT2 glucose transporter to be transported to the blood. So why does it have to be done in the ER? Why not in the cytosol? Well, think about it. Glucose enters, sometimes exits the cell. What would happen if glucose 6-phosphatase is in the cytosol and glucose now enters the cell? The glucose will immediately be phosphorylated by the hexokinase. But then what will glucose 6-phosphatase do? It will dephosphorylate it. So basically, hexokinase and glucose 6-phosphatase will compete constantly with each other. One will phosphorylate, the other one dephosphorylate. The other one phosphorylate, the other one dephosphorylate. So what will be the end product of all this? Just generation of heat. We will be spending ATP, and then we will be releasing that energy from ATP as heat. No purpose. So for that reason, we have to compartmentalize certain reactions. Hexokinase and glycolysis reactions stay outside. The dephosphorylation of glucose 6-phosphate has to be in the ER, controlled by a transporter, so that Only when it's needed, it will be dephosphorylated and then exported out into the blood. Alright, so we see glycogen phosphorylase removes all glucose from the non-reducing ends, and then glucose 1-phosphate is converted to glucose 6-phosphate, and then it can either enter the degradation pathways of glycolysis and pentose phosphate pathway, or be exported as glucose in the blood. Alright, but we're not dealing just with linear polymers here. As you know, glycogen has branches. And how do we deal with these branches? Well, we have glycogen phosphorylase removing one at a time from the non-reducing ends, the moieties, but at some point, approximately 4 residues from the branch, then we have the debranching enzyme that comes in, removes 3 of the 4 in this particular case, places them on another chain, whether it's going to be the same or a different one, doesn't really matter, and then with alpha 1 to 6 glycosidase activity removes the alpha 1 to 6 O-glycosidic bond between these two glucose residues, and we end up with a linear chain. So that's how we handle the branches in glycogen. Now let's talk about glycogen synthesis. Now the first question that I want to ask you is, can glucose spontaneously polymerize into glycogen? And you're probably going to tell me, no. I mean, everybody knows that if you just take glucose and you mix it together, it's not going to become glycogen. The question is, why? Why do you think it will not spontaneously polymerize? And the answer to that would be, because it's perfectly fine by itself. The molecule is stable, it doesn't need to react, it doesn't have any active groups that are highly destabilized and in a need for reaction with something else. The molecule is perfectly happy as it is, and it doesn't feel the need to react with anything. The molecule is stable. So how would you prepare then a stable substrate for a bond formation reaction? What would you do? What needs to happen? Well, if the molecule is stable, which prevents it from polymerizing, then the natural thing to do is to disturb it in a way, or destabilize it, or activate it. In essence, invest energy to it so that then we can use that energy for work. So that is the principle behind the activation of a molecule. Invest energy, destabilize, make the molecule higher in energy so that it will react with something else, whatever that may be, in consequence reactions. And indeed, that's exactly what happens. The way the glucose molecules are activated is by adding to them a nucleotide. So if we name these structures glucose nucleotides, or UDP glucose, because the nucleotide added to them is uridine diphosphate. By adding now this nucleotide, this glucose molecule is activated and ready to be used for glycogen synthesis. So what is the role, what is the benefit of adding these nucleotides to glucose? Well, one thing to say is that their synthesis is irreversible. In other words, once you make it, it stays this way. So what energy change would you expect for reaction that is irreversible? If you pay close attention to what we've talked before, you should say, well, negative energy change, because it's irreversible. But interestingly, here it is. This is the sugar phosphate, this is the NTP, and this is now the UDP, basically glucose, and this is a pyrophosphate, which is the other product. The overall energy of this reaction is small and positive. Interesting. Why is then the reaction irreversible? It's not connected with a large negative energy. If anything, it's small and it's positive. Basically, it suggests that this reaction is just one of those reactions that is close to equilibrium, and just a little bit of energy changes, and so you can easily push it in either direction. That may be the case, but there is something else that happens after these products are formed. Pyrophosphate is fairly unstable by itself. And I shouldn't say unstable, but so much it doesn't really remain for a long time in this form, because inorganic pyrophosphatase quickly degrades all pyrophosphates into inorganic phosphates. Now that reaction has a delta G of negative 19.2, and that reaction is favorable, irreversible towards inorganic phosphate. Now that is the reason why the overall process is irreversible, because of this reaction, the conversion of pyrophosphate to inorganic phosphate. Now what's the second benefit? That because the reaction is irreversible, and now all the nucleotides that are labeled remain labeled, the second benefit is that once they're labeled as glucose nucleotides, they're designated for the synthesis of, for synthetic purposes in general. In this case, glycogen. Another benefit is that, look at this nucleotide. It's even larger than the glucose molecules itself. That extra surface that the nucleotide adds also provides binding surface with the active site. Providing more binding surface with the active site allows for better binding with the enzyme and lowers the activation energy for the catalytic conversion, or for the addition later on of this glucose to the glycogen molecule, growing glycogen molecule. And the last benefit is that the nucleotides are generally excellent leaving groups. So what did we do? Yes, we spent energy here, but we added an excellent leaving group. We made this reaction, this reaction is irreversible, and once the UDP glucose or the glucose nucleotide is generated, it's going to be used for the synthesis of glycogen. And the added also benefit is that the extra nucleotide provides a binding surface for lowering the activation energy. Alright, so now let's talk about now how is glycogen actually synthesized, the mechanism. Well, we're going to start with glucose, which as soon as it enters the cell, it gets converted to glucose 6-phosphate. And then that glucose 6-phosphate is converted to glucose 1-phosphate. Again, it's a familiar reaction, phosphoglucomutase, just working in the reverse way. Now the resulting glucose 1-phosphate is then converted to UDP glucose. And then UDP glucose is now the substrate for glycogen synthase that uses it to add that activated glucose now to the growing chain, or the growing glycogen chain, and UDP here is released. Now, we'll talk about this later, but just to mention it so that, because it's important, is glycogen synthase cannot just simply start adding UDP glucose to another glucose. Glycogen synthase needs to have, or this process happens only when we have minimum of 8 glucose residues bound to each other. That's when this reaction starts occurring. You already have to have 8 glucose residues linked to each other in a chain. When we have that, that's when glycogen synthase will bind to it, and that's when glycogen synthase will start adding the new UDP glucose molecules to the non-reducing ends of that minimum 8 glucose residues chain. Alright, so now we have to answer again the question, what about branches? Each beta particle has about 2,000 branches. How will those happen? Right here we're looking at the extension of a linear chain, but there's no branching. Well, we have the branching enzyme, which when we have an approximately 11 or so residues bound into a chain, then the branching enzyme comes, takes the last 6, 7 residues and binds them to the 6th carbon of an internal residue, the 2, 3 residues from the cutoff site, and adds them there. And then the process continues to extend, and then the branching continues to happen whenever we have about 11 or so residues long chains. Now, again, a good question would be, why do we want to have branches? What's the benefit? Why would the cell want to spend energy and resource to have even specific enzyme to actually make branches? Why can't we just continue with the linear chain and just keep going on and on and on? Well, obviously, I don't know the real reasons for that, but I can tell you how branching benefits synthesis and degradation. Remember, each one of those branches ends with a non-reducing end. Synthesis or addition of new glucose molecules or the removal of glucose molecules during the degradation happens only at non-reducing ends. So by making branches, now we went from one end to 2 non-reducing ends. Now instead of 1 glycogen synthase, we have now 2. So the rate of synthesis will increase. And then the new branch, and the new branch, and the new branch, and soon we'll have 100, 200, 1,000. And each one of those ends now represents a binding surface where a glycogen synthase a copy of the glycogen synthase enzyme can bind and continue to add more glucose faster. On the opposite, glycogen phosphorylase can add now can bind to each one of those ends, and instead of removing one at a time every single time from a linear chain, because the enzyme can only break one at a time, at the same amount of time, if you have 2,000 non-reducing ends with 2,000 copies of glycogen phosphorylase, you can generate 2,000 glucose molecules for the same time. One will be removed from just a linear polymer of glucose, such as amylose. So that, I think, is the benefit of having branches. Faster synthesis, the more the branches, the faster it happens, and faster degradation. The more the branches, faster degradation occurs. Alright, so I told you that glycogen synthase cannot start simply to add glucose to or one glucose to another. In order for glycogen synthase to start working and generate the full molecule of glycogen, it needs to have a so-called glucose primer, which is generated by at least 8 glucose residues. Well, obviously the question would be, well, if glycogen synthase doesn't generate that primer, but it needs it, then who or what generates that primer? There is a protein that has that ability to catalyze the first reaction and then the addition of 7 more glucose molecules and generate the 8 glucose residues primer. That protein is known as glycogenin. It's shown right here. Glycogenin has the ability to bind UDP glucose and to add the first glucose residue to its tyrosine 194, to its hydroxyl group of tyrosine 194. Then that reaction, once we have the first addition, then that reaction repeats 7 more times to form the primer of 8 glucose residues bound into a linear chain. This is the reducing end of glucose, of glycogen. There is only one reducing end, and that reducing end is bound to glycogenin, shown right here. And then branches will occur more branches, etc., etc., and that's how we form the beta particle and the alpha rosettes, and then the storage of glycogen occurs in the hepatocytes or myocytes. Once we have the primer generated, then glycogen synthase binds to it and starts to add more glucose, and then the branching enzyme, like I said, comes in and generates branches, etc., etc. And with that, I conclude everything I have planned to talk about during chapter 15. I hope things are clear. If they're not, please let me know. Come and see me during my office hours, or email me. I'll be happy to answer any questions. Thank you.